the Pro-Chancellor, Mr. Winston Thompson, and colleagues, and students, undergrad, postgrad. Let me say to you all a very warm welcome, and also to thank you for agreeing to participate in presenting your research work that you are doing at different levels. In fact, originally we were thinking of only having the postgraduate students, like masters and PhD, to make a presentation. But later on, in our discussions for the campus celebrations, we agreed that we should give the opportunity to undergraduate students too. Now, I like, for the information of the Pro-Chancellor, I think we are the only school that we have a course, AG 383, where we teach students, undergraduate students, how to go about conducting a research. So they learn all aspects of preparing a research proposals under the guidance. There is no formal course as such, but they go through from time to time theoretical, maybe a week during a semester or so, but it's done at different times to make sure that the student understand what research actually involves. You have to have a topic, you have to have a methodology and research design and literature review, and then how you're going to report your findings, data, and then your explanations or descriptions and conclusions, recommendations. All of those things have to be considered. So the student who, before, students will complete the degree, but when they go back home, they say, okay, the management says, okay, can you do research on taro or yams or whatever, whether it's plant protection or agronomy or animal science. The student knew about those things, but they did not know how to objectively design an experiment and carry out the results and then recommend that this is what we found, and anybody else, wherever they repeat this research, they will get the same result. So now our students, I'm very proud to say that many of the research data that was collected by the students have actually been published in journals which are ranked together with the staff who are actually supervising the students. So the research, the level of research is really high. The quality of research is very high. It's just like somebody who is a researcher, already qualified, whereas these students are still continuing their undergrad degree. So I'm very proud to say that some of your research have been excellent, and then you will have a good feel for it when they have finished their presentations. And for masters, again, it's a small school, but we have had a number of, in fact, we had the largest number of graduates in master's degree last year among all schools of FBE. And this year, we'll also have several students who will graduate, including possibly 2 PhDs also. So it's a very good record for us, but then we have to share that information. And I will now hand over to the research coordinator, who is actually managing the research activity together with other staff to please organize the actual presentations. Thank you very much, and thank you one more time, Pro-Chancellor, Mr. Winston Thompson, for making yourself available. And I should say too, for your information of all of you who come from different countries, this is the first Pro-Chancellor who actually comes to this kind of things. Others, they will probably be somewhere else, meeting and doing. He takes a lot of interest. And, the very good thing about him being here, he will know the problems we face. He's the chair of the council of the university, the highest governing body of the university. He chairs it. Now he will know firsthand, no second-hand information. You're going to say, this is what I am suffering from? Tell him. He will be very happy to know. And then we can only improve after that. We can't go down, because once something is a problem, we can only go up by improving it. Thank you very much.
Good afternoon, everyone. Welcome to our presentation this morning. Our team will present to you on the topic, the effects of vegetative pruning on the yield of food yam, Dioscorea pneumalaria. We have our outline in front of you. Yam propagation in farmers' field use tubers. But the problem is up to 50% of tuber production is used for planting material. That's the problem. And it raised the question, is there any other way which we can use so that we can preserve that 50% for home consumption and market as income for our farmers? Vine cutting is an alternative for that. How are we going to do it? You can see from the diagram in front of you. The first diagram is prune the vine, cut them as planting material. Each planting stock should have at least 3 nodes. Nursery in the garden, in the nursery house by using poly bags and soil. And the third diagram is, by then it should develop root system. That is the right time for you to transplant it out in the field. And 5 months time, you'll have the production, as you see in the front. The problem is, up until today, no information on the effects of pruning on the tuber yields. Aim of experiment, to study any effects of vegetative pruning on tuber yield of yams planted for mini sets. Our objectives for our research, number 1, is to study the growth and tuber yield responses of mini set planted yams to differentiate levels of vegetative pruning. Number 2, to determine the effects of pruning on the eating quality of the tubers.
Methodology. The experiment was conducted at Vaitelauta. The mini-set propagated yam plants were planted 1.5 meter between plants, 2 meter between rows in a randomized complete block design with 4 treatments and 5 replicates. The following percentages of pruning were used as the 4 treatments. Treatment 1, 0%, treatment 2, 25%, treatment 3, 50%, and treatment 4, 75%. For data collection, Fig 1 shows the pruning applied about 2 months after planting. Fig 2, leaf area index, measurement done monthly using a canopy analyzer. Fig 3 shows the harvesting was done at 7 months after planting. And Fig 4 shows the weighing of tubers done after harvesting.
Looking at our results and discussion, from the graph you can see the leaf area index. The first 1 month is from the vegetative to the second month. When it comes to the third month, you see the highest reproduction of the plant area index. So from there you can see for the 4th month, the plant becomes senensis. Senensis shows that the plant's being ready for harvested. In here, you can see the tubers healed after 7 months. You can see that the yield for 0% control, 25% control, 50% control, and 75% control. Looking at the graph, it shows that the 0 and the 25% are comparable, and so 50 and 75%. But there is a significant difference compared to 75%, 0, and 25 in terms of yield. Our samples were analyzed in the lab. This is the analysis of the dry matter. In the graph, you can show that the 0% has the highest dry matter content compared to 25, 50, and 75%. It indicates that the higher dry matter content, then the better eating quality.
For our conclusion, vegetative pruning at 25% and 50% produce the same yield as the unpruned plants. Extreme vegetative pruning at 75% reduces yield. Pruning yam reduces tuber dry matter content, therefore eating quality depending on eating preferences. For our recommendations, pruning should be done below 50%. Do not prune yams if hard texture of tuber is valued as an eating quality. Thorough minimum tillage to be used when experiment is repeated. And finally, encourage farmers to use vine cutting as an alternative method for planting material.
Where to from here? In our research requirements, fine-tune vine cutting technology. Second is study appropriate management of vine cutting propagated yams in the field. As you can see from the picture, that vine cutting propagated can produce tuber of up to 65 grams in 5 months. Remember that one vine cutting can produce 1 up to 4 tubers. So what is the benefit and the possibilities? 1, source an alternative planting material. 2, baby yams can be a new agricultural produce on the market for monetary return for family. It is also a potential import substitute for Irish potato. This is our reference. Thank you for listening.
Popular variety used here, or is there another variety that is the more popular one in the market?
This is a popular variety used here in Samoa, as well as other Pacific countries. So we see it fit for us to take the research on.
Very good afternoon to you all, and welcome to our presentation. We will be presenting on the influence of peeling and calcium level on the utilizations of giant dallo, also known as calcium macrolizer cornmeal, by laying hens. Feed is a limiting factor to improve or increase the production of poultry here in the South Pacific Islands, whereby we need more research into the feeding value of local resources. Giant dallo or calcium macrolizer is a readily available root crops here in the South Pacific Islands. According to researchers in 1995, due to its high calcium oxalate, it reduced the performance of poultry productions. However, addition of inorganic calcium, it helps to overcome or reduce the adverse effects of calcium oxalate, according to that source. Objectives: to combat the effects of peeling and calcium level on the utilizations of giant dallo, cornmeal by laying hens. Specific objectives is to study the effects on egg productions, feed utilizations, egg quality, both internal and external.
Methodology. The research was conducted at the Areta Poultry Unit. First, we purchased the giant taro, and then we divided it into 2 parts, peeled and unpeeled, and then both were chopped into small pieces and sun-dried, and then later it was grinded for feed formulation. The feed was then formulated into 6 different diets. Diet 1 had negative control, where we did not include giant taro meal, but calcium carbonate was included at 3%. Diet 2 was our positive control. Again, it did not include giant taro meal, but it had calcium carbonate added at 4%. Diet 3 and 4, it had 15% giant taro meal included, peeled and unpeeled at 3% calcium carbonate included, while diet 5 and 6, it again included 15% giant taro meal, peeled and unpeeled at 4% calcium carbonate included. And then each diet was replicated 3 times and then fed to 5 birds per replicate.
Results and discussion. As you see, graph 1 shows the laying performance by Sever Brown, fed, peeled and unpeeled giant taro with different levels of limestone. The result shows that there's no significant difference in the parameters feed intake, egg weight, hen day production, egg mass, and the FCR, which is feed conversion ratio. These results are similar to Oikali et al. 2016, which found that inclusion of giant taro at 20% level, there's no significant difference in the laying performance. Graph 2 shows egg qualities of Sheva Brown, fed, peeled, and unpeeled giant taro with different levels of limestone. This result shows that there's no significant difference in the parameters, shape index, shell thickness, hoof unit, and yolk color. These results are similar to Oikali et al. 2016, which also find at the 20% level of giant taro inclusion has no significant difference in egg qualities. Conclusion, at 15% inclusion of giant taro root meal, peeling or additional calcium is not required. This would be beneficial as peeling would be an additional cost. More research into higher levels of giant taro and calcium level is recommended. Thank you.
You are saying that the weight of the egg was up to 60 kilogram. It is a gram or kilogram? I never seen such a heavy eggs of any bird.
Yeah, it's gram. Sorry, there's an error.
You were too busy and you forgot to put that. Okay, any other queries? Okay, please put your hands together again for our group.
Alafua, my name is Tanya Lesa, and the topic for my research is the effect of irrigation interval on growth, yield, and water use of lettuce. Lettuce is one of the most imported vegetables in Samoa. In 2016, the trade statistics showed that Samoa spent $7 million in importing vegetables, of which lettuce was the third most highest. Lettuce is very popular in Samoa because of its nutritious value, and the demand from the local supermarkets, the hotels, and the resorts are not being met by the local farmers. One reason being is that they are not able to farm lettuce throughout the whole year because of weather conditions. So the Ministry of Agriculture and the local farmers have been looking for ways that they can increase yield and farm lettuce throughout the whole year. The answer to that was tunnel house and the drip irrigation system. Research showed that lettuce that were grown in the tunnel house produced more yields than lettuce that were grown out in the field. And they were able to grow lettuce throughout the whole year. By using the drip irrigation system, they were able to save water and cut down on production costs. So this research wants to go a step further and try to see if we can reduce the amount of water that is irrigated to the plots, but still produce the same high yields. So the objective of this research was to determine the effect of different irrigation intervals on the growth and yield of lettuce. Also to determine the water use efficiency, and from there develop irrigation management recommendations for the farmers. The experiment was conducted in two locations. One was the Nu'u Research Station, and the other was a rural village, Sa'anapu, and the tunnel houses were used. That was the experimental design in the Nu'u field and the Sa'anapu field. So 4 treatments were used. Every time irrigation was done, 4 millimeters of water was added to the plants. So the first treatment was daily irrigation. Plants were irrigated every day. The second treatment, plants were irrigated every 2 days. The third treatment, plants were irrigated every 3 days. And the last one, plants were irrigated every 4 days. So these were the growth and yield parameters that I collected. And the result shows that irrigation interval did significantly affect 3 of the yield components, which was the plant weight. As you can see, the 2 days interval produced the highest plant weights compared to the other 3. For the head diameters, the 2 days interval also produced the largest heads compared to the other 3. For leaf number, the 2 days interval also produced more leaves than the rest of the treatments. This graph shows the relationship between crop yield and the irrigation treatment. The blue bars are the amount of irrigation water that was added into the plots, and the red dots show the amount of yield that were produced. So you can see with the daily irrigation, so much water was used, but that was how much yield it produced. But with the 2 days irrigation, not so much water was used, but it was able to produce a very high yield. This graph shows the relationship of water use efficiency between all the irrigation treatments. And the lowest water use efficiency was in the daily irrigation because it used so much water, but it produced not so much yield. So the conclusion of my study, the irrigation interval significantly affected some yield components of lettuce. They were fresh weight, head diameter, and leaf number. It also significantly affected water use efficiency. And we found that the 2 days irrigation interval treatment produced the best yields out of the 4 treatments. So the recommendations for the farmers that are using the tunnel house and the irrigation system is they should use the 2 days interval because they will not only produce much higher yields, they will save water, save labor, and definitely save money. So with that, ladies and gentlemen, thank you very much.
Good afternoon, everyone. Welcome to my presentation. This is the result of my master's research, and the topic of my research is characterization of elite cocoa trees in Makira Island, in Makira Ulawa Province in the Solomon Islands. As you all know, my name is Ellison Toramo. I'm from Solomon Islands. My principal supervisor is Dr. Desai and co-supervisor Mr. Falnico. By way of introduction, cocoa is an introduced crop in the Solomon Islands, and the first introduced types of cocoa that were brought into the country composed of Trinitario, Criollo, hybrids, as well as Amelonando types. And it was between the period of 1950 to 1960, respectively. And the farmers were growing a mixture of those. And in 2013, the Ministry of Agriculture and Livestock in the country, they released an information that the cocoa variety in Solomon Islands is highly variable. However, there is no scientific proof or scientific evidence for that particular statement. And so the information that is generated from this research will be very useful in terms of designing future breeding programs for finding good varieties for the farmers to grow. I have 2 major objectives. The first one is to identify the morphological characteristics of elite cocoa trees in Makira Island, and the second is to determine the genotypes of those elite cocoa trees. This is the study site that I went through during the data collection. As you all know, Solomon Islands has 9 provinces. There are 3 main cocoa growing provinces. And the study was conducted in the, you can see in the red circle, that map shows the location of the island. And the map on the other side is showing the island and the location, the geographical location of the elite cocoa trees that were sampled. And the study was carried out in wards 7, 8, 10, and 11. As part of the methodology, this is what I did in the field. Figure 3 to figure 8, they are involving morphological data collection. It was collection of data for the morphology. And then figure 9 and 10, that's I collected the young leaf buds, and I packed them and I sent them to the Asia counterparts for DNA analysis, DNA extraction, and as well as analysis. Here's the result. The first result that I have is I also went into analyzing the quantitative traits in the words, among the words, and you can see the traits that have significant difference are the ones in color. You can see, and the rest, there's no significant difference. So you can see the port external thickness, the port internal thickness, and the cotyledon length. Those are the only ones that they have significant difference. That same information was converted into graph form, you can see here, so that you can be able to easily tell the port external thickness and the cotyledon length, as well as the port internal thickness. Those were the differences, the significant differences. So the numbers here, 7, 8, 10, 11 are the words. In other words, they are geographical locations where the farmers were, but they are residing. Here's another analysis I did. This one is a dendrogram showing tree clusters. I did this one based on the phenotypes. 18 characters were clustered together, and then I came out with 3 clusters. In other words, 3 groups. So group 1, you can see it's from cluster 1. The green line, it's a very, very long line. That's the majority of the pods or the samples. They were classified in group 1, and group 1 you can see here. And group 2, very few. Group 3, very, very less. So group 1 is the most. The cluster 1 makes the most of the samples or the trees. And another analysis here is the principal coordinates analysis of the genotypes. Previously it was the phenotypes, and these ones are the genotypes. So you can see here on the slide different colors. The Solomon Island samples, which I did, they're the ones in red. So you can see in the red, they are circling around Amilonando. They are circling around Amilonando. So this analysis tells us the relationship of the samples with the reference clones. So we can see here it's only one is sitting right here, meaning that it's quite different from the rest. So on the side of the table there, I put them in red just for visibility, that Amilonando is popular, while those ones in red, they have reduced Amilonando traits in the samples. So you can see for yourself. So they made up of other different varieties as well. So that's what the research found in this particular studies. By way of conclusion, I think it's good to link the phenotypes and the genotypes as well. There's no slide showing that. You realize from the cluster analysis that the bulk of the samples, they were in cluster 1. And so also in the genotypes, the bulk of it is Amilonando. So this studies has the major findings that the phenotypes and the genotypes, they correspond. They correspond. They agree. So the conclusion is that the study found the cocoa population in Makira Island of Makira Ulao province of the Solomon Islands as mainly consist of Amilonando type of cocoa. And also some elite cocoa trees, as we can see in the table in the previous slide, they're also having some DNA strains of Criollo, Parinari, IMC, Nanai, and the national types of cocoa. These ones are the different types of cocoa varieties. And thank you all for listening. Thank you, Thomas.
Thank you, Alison. The floor. Yeah.
Cocoa is still a significant crop in that region? Cocoa is still significant as a production?
Thank you, sir. Yes, cocoa is currently is an export crop in the Solomon Islands, and the value range between 80 to 100 million annually. That's SBD, Solomon Island dollar. It goes with the production each year, but that it goes up just between that range.
Is it a lifeline crop of the island? How many percentage of the population is engaged in the production of cocoa?
It's around 133,000 families that they engage in cocoa growing. Currently, that's countrywide, all.
Is there a lot of cross pollination between Melanado and the others? Cross breeding.
As we can see in the result, very little. Yeah, that side here. The ones with the color is the ones that they cross-pollinate. It's a natural hybridization in the field. Yeah, so those are the ones. Otherwise, you can see the Amilonando is ranging. The marked ones, they are the ones that they fall just right from 53.7 downwards. Amilonando. The rest, they are above. So that is the level of cross-pollination we can see.
You are measuring the pod diameters, and then you go back. You are measuring the inner and outer diameter of the kernel of the pod. So that's how much
This one here is measuring the pod circumference. Which one? This one? This up? That side? Oh. Thank you for your question, sir. The procedure is only covering some. It's not all. So this one is also part of the procedure I followed. Also the external and the internal thickness. The internal or the thickness of the pod is also giving some indicators of the bean size.